Okay, guys, so here's the deal. My daycare provider has the flu, and daycare isn't open, so I'm at home with Riker today. What we were going to do is talk about DNA replication again, or at least in a little bit more detail, finishing up with the Chapter 13 notes that you all have access to on Canvas today, and then use those foam boards that I promised you. Um, so you don't have access to the boards today. But I promise I will get them out for you when I come back. I should, by all means, be back tomorrow. Um, so here's the thing: DNA replication, like we talked about today in class, is important for leading into mitosis and meiosis because, as you recall, there's an interphase with an S phase, S standing for synthesis, and that's where the DNA is duplicated before mitosis as well as before meiosis. Okay, so there's DNA replication, and that's strictly for that reason. Um, so, if we're concerned about what you need to know for that, you just really need to understand how basically that DNA or genetic information is copied um, for basically transmission between generations, so passing on that DNA from one generation to the next, whether that be in mitotic division from a parent cell to a daughter cell or meiosis and creating gametes and then further on offspring. Enzymes are really important. And like I said yesterday in class, some of those enzymes can get a little bit confusing because they sound similar to the ones that we're going to talk about when we look at protein synthesis. So what we're going to do now is, as you can see on the screen here, I've got the notes, the chapter 13 notes up here. We're going to finish those. So there's a few of them here. Basically, we're going to run through the steps of DNA replication. I also have a video that I'm going to play. It's like 3 minutes long. So we're going to play the video. We're going to listen to it together. I'm going to stop it and, I don't know, add my 2 cents in. And then I'll tell you what I want you to do afterward. On the AP biology exam, there may be some questions on DNA replication, just that topic. Usually they try and maybe compare and contrast DNA replication, to protein synthesis. Or perhaps they're going to ask you, in lieu of evolution. They love that evolution tie-in. So we'll add some more layers to this, but what I got from class today was that you guys really need a good review on DNA replication. That's not the meat and potatoes of the AP biology exam, though. What they're going to focus on is protein synthesis, the central dogma, DNA to RNA to protein. So that is where we have to spend the majority of our time. So this is basically it for DNA replication. And then if you have questions beyond that, I encourage you to read in your textbook or come in to me during Brave Tutorial, ask me questions during class, when we're using those foam boards in the next couple of days. But we are going to spend the majority of our time on protein synthesis coming up. Okay, so that's my plan for starting on Wednesday, tomorrow. All right, so anyway, okay, fast forward through all that jazz now that I made you listen. This here, I'm going to keep it like this on my screen. I hope that's not too much of a bother. If it is, suck it up. We've got our parental DNA, and as you know, it's a double helix, right? It's very tightly wound. There's an enzyme that works on this side of the unwinding DNA molecule, and that is an enzyme that kind of takes the strain off of things. So if you are wearing a sweatshirt and you've got your hoodie ties, and you start rolling those up, you know how it gets tighter and tighter and tighter towards one of those ends? That's basically what the DNA strand does. So we need the enzyme topoisomerase in order to kind of lessen that strain. Okay, so I don't have a very good picture right now. Sorry, guys. Oh, here. Yes, I do. This guy here. So topoisomerase is going to be that enzyme that lessens the strain on that full parental strand of DNA. So there's no breakage or damage to any of those covalent bonds holding together our backbone. Okay? So that's one really important enzyme. This slide here goes through the major steps and it talks about those enzymes. But I'm going to go back to this picture here. So imagine topoisomerase out here at the front. We know that DNA is anti-parallel, so this strand runs 5' to 3', whereas the opposite end, 3' to 5'. Now, here's the tricky thing that students sometimes get confused. When the new strand of DNA is synthesized, it synthesizes the new strand in the direction 5' to 3', meaning that the template strand is actually 3' to 5'. That's confusing. The new strand of DNA is synthesized in the 5' to 3' direction, okay? We've got two different strands working here. Since we've got that anti-parallelism, we've got a leading strand. This leading strand is synthesized in a continuous motion from start to finish. Then we have this lagging strand. So if you notice, that lagging strand, the template, is running 5' to 3'. So basically what this strand has to do is synthesize these short fragments in that 5' to 3' motion that it likes, but it's basically like I think of trying to swim up current on a river. Okay, it's harder. The enzyme has to do more work. So there's a little bit more detail in this lagging strand as to how it will actually make a copy of that DNA. Now, that's not to say that it doesn't happen, because obviously it does, but just know that there is a difference between the leading, which is continuously synthesized, and the lagging, which is synthesized in fragments. And they're synthesized in fragments that are called, I say, Okazaki fragments, Okazaki fragments. Some people pronounce it differently. It's named after the scientist that discovered this phenomenon, okay? And then those fragments, like you see here, here's one, could be another one. Those fragments are going to be joined together with the help of another enzyme that we're going to talk about, okay? So our major enzymes are that topoisomerase out here trying to create less strain on our parental DNA molecule. The other one that's really important is DNA polymerase 3. This does the major work in pairing up new nucleotides to make that new strand of DNA, okay? Another enzyme that's important is helicase. Helicase works right out front in order to unzip this DNA molecule and open it up. Keep in mind we've got those weak hydrogen bonds that are holding our bases together. So helicase is going to break those enzymes. It's going to unwind the double helix form of this DNA. Okay, so it works ahead of this enzyme DNA polymerase 3, obviously, because we have to open up the strand in order to get at it and add new nucleotides. These thingies here, these gray doodads, those are called single, how do they phrase them in your notes? Single strand binding proteins, I think. Basically what those are there for is support, okay? What they're going to do, since this parental strand of DNA is now completely open, it's not supported as well. There's no van der Waals interactions trying to hold those bases together, nor are the hydrogen bonds intact. So these single band protein enzymes, I can't even think straight here. Sorry guys, hold on. Single strand binding protein. My gosh. Okay, fast forward through that indefinite pause there, please. Single strand binding proteins. All they're there is for stabilization and support, okay, of these two open strands, template strands of DNA, okay? All right, another DNA polymerase 3. Again, it's working in that 5' to 3' direction, but it's on the lagging strand, so we're synthesizing in little bursts. Another enzyme that you're going to see is a DNA polymerase 1, and that works on this lagging strand to take out these primers that we talk about. Those primers are put into place by something called primase, and they're RNA primers, in fact. So RNA is different from DNA in a few different ways, but one of the main things that we talked about in class is that RNA doesn't have thymine, it has uracil. And so primase comes in and puts these RNA primers on both the lagging strand and the leading. Now, the leading strand only needs one primer in order to start the process of DNA replication and then finish, since it's a continuous synthesis. The lagging strand, since it's synthesized in these short little fragments, there are numerous primers that are necessary. So primase puts the RNA primer on the strand, signaling, hey, start here. And then DNA polymerase 1, when everything is pretty much said and done, is going to come through and remove that primer and then replace the nucleotides with DNA. So essentially what it's doing is it's taking out that RNA uracil and putting in DNA thymine. Okay? Last enzyme. Very important, it links everything together. It's that DNA ligase for linking. This links together those Okazaki fragments and then makes sure that those hydrogen bonds are intact on both the leading strand and the lagging to synthesize a complete double helical DNA strand now. So, as we said in class yesterday, it's a semi-conservative replication. So focus on this right here. Semi-conservative meaning that once this parental strand is opened up, DNA replication occurs, we end up with one parental strand and then one new strand, okay? One new, one old. All right. Again, this gives you kind of a synopsis of what I just told you in all my great wisdom, and then this breaks it down by certain steps. Helicase, of course, does the unwinding, and what it does is it forms these replication forks. So we're going to look at the difference between DNA replication as well as protein synthesis, which will focus mostly on protein synthesis, between prokaryotic organisms and eukaryotic organisms. Prokaryotic organisms, such as this E. coli bacteria, their DNA is circular. So when DNA replication occurs, they only have one replication fork. Okay? There's only one. See? Only one. DNA then is replicated, and we end up with two strands of DNA. Again, it's still semi-conservative, okay? Or two full molecules of DNA, I should say, not two strands. In eukaryotic DNA, since it's linear, you notice that we have 1, 2, 3 replication forks in this instance, okay? So in eukaryotic DNA, due to its linear form, it can have numerous replication forks. Why would that be important? Faster DNA replication, okay? Let's see. This one talks about primase. Like I said, puts that RNA primer on, saying, hey, start here. Oh my goodness. Okay, this goes through leading versus lagging. Some students don't like these notes here because they feel like it's a little too confusing. So if it confuses you, please don't look at them. Only look at things that help you, okay? This is just going to talk to us about, when we, again, synthesize that new strand, the new strand runs in a 5' to 3' direction, okay? One, the leading strand is continuous. The lagging strand synthesized in these short little bursts of fragments, okay? All right, more info on summarizing those enzymes. You'll notice that we talk about these polymerases and the primases when we discuss protein synthesis, so it's really important to keep these straight. I had mentioned maybe making flashcards, a stack for DNA replication, and then we can make a stack for transcription and translation in protein synthesis. Not a bad idea. Okay. The other thing that I want to touch on is the fact that with our bases, adenine always has to pair to thymine, cytosine always has to pair to guanine. Okay. How does this work all the time perfectly? It doesn't, okay? I think we would probably be surprised by how many errors there are. This is a fairly fast process. It's complicated, especially on that lagging strand. But our DNA, our whole genetic makeup has basically made itself a proofreading factory as it's going through and doing its DNA replication job, okay? You'll see the proofreading is happening in protein synthesis as well. But DNA polymerases, even though they are adding those nucleotides, okay, so they're supposed to be, if they see an adenine, they're supposed to add a thymine, right? Well, what if they tag on a guanine instead? The DNA polymerase is not only doing its job of adding those nucleotides in, but it's also kind of like a spell check in our Word documents or autocorrect on texting. What it's going to do is it's going to make sure that the proper bases are paired accordingly. So usually there are some errors that occur. A mismatch repair, that means that there are special enzymes that will come in and fix incorrectly paired nucleotides. So it's flagged by that DNA polymerase. We also have something that, let's see right here, nucleotide excision repair. So what happens is that this nuclease, this enzyme, is going to cut out any damaged DNA, and then DNA polymerase is going to fix that damaged DNA, pair it up with the correct bases, and then ligase is going to come in and do its linking job all over again. Okay? So that's kind of a backup to any problems that may arise. So if DNA polymerase can only add these nucleotides in a 5' to 3' direction, meaning that it's reading the parental strand in a 3' to 5' direction, it means that every time a chromosome is duplicated for the process of mitosis or meiosis, there could be a very small portion of the tip of the chromosome that is removed. Okay, so in that lagging strand, it's not always going to go to the end, right? It's not a continuous, full replication of that strand. It is replicated in those short fragments. Well, what if there's no primer at the end to signify a start here? So there are these caps that are on the end of chromosomes. They're called telomeres. Okay, telomeres. Those telomeres are really, really important because they're a cushion, basically. Those telomeres, what they're going to do is they are going to basically protect the end of the DNA that could be lost or degraded in that process of replication for mitosis. Okay, so yes, we do lose a little bit of those chromosome tips every time that our cells go through mitosis. However, we have this backup plan, and that's the telomeres. And it's these short little fragments of repeating sequences of nucleotides. So say this is one sequence that's there, you would see maybe 50 others, okay? So if we end up losing this segment, whatever, we have 49 left, okay? So we still could have 49 mitotic divisions before we really have to worry about our chromosomes being damaged. So what this essentially means is that any given cell has basically a certain number, or finite number of times that it could possibly divide before we start to lose really, really important information on that chromosome. Okay, and that can be very detrimental to a person's genetics. In your biology textbook, if you guys remember reading any of that, there was something mentioned about twins, two male twins. One was an astronaut and was sent into outer space and spent like a year there. And what they did was they took a full genome analysis of both of them prior to this one twin rocketing into space, and they checked his telomeres, right? And so when this guy up in space came back down to Earth, then they did a full genetic analysis on both of them, and they realized that the twin that had actually gone into space, his telomeres had started to lengthen, and the one that was here on Earth for that full year, his telomeres started to shorten. So there was some research in that, I guess. I don't know. Anyway, fun fact. Another fun fact. Let's talk about tumor cells. If you've ever read the book about Henrietta Lacks, she was this African American woman who had a form of leukemia, and her leukemic cells are still within research labs to this day. They're actually frozen, and they're the primary source of research for that particular form of leukemia. Her cells are, in fact, cells that I did research on when I worked in the leukemia research lab up at Minot State. So these tumor cells, what they have essentially is this mutation. And what that does is it activates this enzyme right here, telomerase. And that enzyme is going to add to those telomeres. So basically, there's no worry of losing essential information. Now, that would be nice if it wasn't a cancerous cell, but that's not the case. So, research is going into trying to figure out if this telomerase could be used in regular, healthy functioning cells. All right, this video here doesn't play on my computer, it won't play on your iPads either because it needs Adobe Flash or some silly thing like that. So here's this video. It's about 3.5 minutes long. All I typed in was DNA replication animation. This is a video that I use in biology and AP Bio. So
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strand.
Keep in mind, when we talked about those replication forks, there's only one in prokaryotic DNA when they go through DNA replication, and there can be multiple within eukaryotic DNA.
Each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will...
Okay, DNA polymerase, yes, but it's DNA polymerase 3. Okay, we have 2 different types of DNA polymerases, so make sure you understand this is DNA polymerase 3.
Make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made...
Hold on. The new strand, 5' to 3', the old strand would be anti-parallel, so the old strand would be 3' to 5'. Okay? Make sure you get that straight in your head.
Continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction.
Look at them go. Continuous.
The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer.
Just like the leading.
DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers.
Exonuclease here that she mentions, yes, but this is the DNA polymerase 1 that I was talking about.
from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so at the beginning, we have our enzyme helicase that comes through and unzips, like you're unzipping your jacket, essentially. Okay, hold on. All right, there's our helicase, okay? Unzipping the DNA, creating this replication fork. We've got our leading strand here because it's running in our 3' to 5', so we can easily synthesize 5' to 3' in a continuous direction. This here is our lagging strand. This is going to synthesize in those short little fragments. So this is especially, This is especially concerning when we talk about those telomeres. Okay, so this lagging strand is what we're concerned about, losing those telomeres at a faster rate than what the leading strand ever would. Okay, so then you've got your DNA polymerase that works because it picks up a signal from this primer that's placed on by primase, telling it to start here, okay? Kind of like you get the address to a party. You're going to go to the party, right? But you guys don't do that, okay? So it just synthesizes continuously, DNA primase 3, nice and easy on that leading strand. Lagging strand is a bit different, okay? So it's going in the 5' to 3' direction, but it takes a little bit more. So it's going to have primers that are added throughout, just giving it a little extra help. So it's going to synthesize in those short little fragments called Okazaki fragments. Then we end up at the end removing that RNA primer, and DNA polymerase 1 is going to replace that primer with the correct DNA bases. Okay. Last thing that happens is that we have our DNA ligase that comes through and links our nucleotides together, specifically at the nitrogenous bases, making sure that those hydrogen bonds are intact between adenine and thymine, two of them, and then cytosine and guanine having 3 between them. So this DNA ligase is essentially you zipping your jacket back up when you go outside, okay? All right, so what I want you to do is go through this again if you found it helpful. Bozeman Science, Khan Academy, watch this video here again. Another thing I might recommend, we've got our amoeba sisters, and although they're cheesy, they get the point across. There's this video here as well that I've watched and I like. This one isn't terrible either. This one here is good, but the animations are a little wild for me, but it's just totally up to you. So watch some of these. Get the DNA replication process down, knowing that it is for that mitotic division, okay? So this is one thing, all right? So knowing the enzymes, what they do, synthesizing 5' to 3' for a new strand, semi-conservative replication. Knowing that there is that proofreading system that's found within the enzymes that help undergo DNA replication. Okay. So what I want you to do now is, once you've gone through some videos, you feel like you have an okay handle on it, I want you to take those 2 handouts that I gave you yesterday in class, the one on transcription and the one on translation. It asks you about like where transcription is occurring in eukaryotes, where is it occurring in prokaryotes, what enzymes come into play, and then it gives you a couple questions on what if this, what if that. So what I want you to do is I want you to make a comparison table of DNA and RNA, and if you've looked at all at the chapter 14 notes that are available to you in PDF form on Canvas, oh gosh, right there, look at that, DNA versus RNA. So maybe you just study that, but like I said, there is something to be said about writing things down. So, what I want you to do is focus on the central dogma, DNA to RNA to protein. So, you're going to fill out that worksheet on transcription. Then you're going to fill out the worksheet on translation. Now, you're going to say, well, you haven't gone through the notes yet. How am I supposed to know this stuff? Well, you should have read. You should know some stuff from biology. You should know that you can open up your textbook and use that while you do these worksheets. And you have notes right here, okay? So you're going to fill those worksheets out. I'm going to come back to you on Wednesday. We're going to talk about these notes. We're going to talk about those worksheets. But if I see blank worksheets or you tell me that you don't have them, then I'm going to come back and I'm going to be angry, okay? You cannot be on your cell phones. You cannot be on some stupid Pinterest or Snapdoodle or whatever the heck you people do. You need to do those worksheets. We don't have a lot of time for this crap, okay? And it's really confusing stuff. So we have to get a handle on it. So you've got to work for me today. Otherwise, I'm going to come back angry. Okay. So look through all of this. Remember things about amino acids, blah, blah, blah. Look, amoeba sisters, okay? Again, watch videos if you like the animations as you fill out those worksheets because they do help. And then, oh gosh, we'll take it from there when I come back. Okay? All right, that's it. How do I stop this thing?